All right, class. So let's go over how to read the hematocrit one more time quickly. So you see the capillary tube, right? So one thing, the bottom of the capillary tube right here, do you see that's the bottom of the capillary tube? You do not align the bottom with 0. No, you align the red mark. You align the red mark where the blood cells are. So you align this where you see the blood cells. You align that with 0. And then the top should align with 100 on the scale, right? And then, as I mentioned before, you just see where the red blood cells are. And if you continue here and read, it's between 40 and 50, so it's about 45%. Okay, again, there is this video, a YouTube video link that I have posted. It also shows you nicely how to read your hematocrit value. I recommend you watch it. I think it's 1 minute or something. It's really short video, and it actually shows you how to read it. All right, so that was about the hematocrit experiment. But now the question is, where are you going to dispose of all these? Well, something to pay attention to. So the hematocrit tubes and the lancets obviously goes in the biohazard sharp. Alcohol swabs, band-aids and other materials that touched blood sample should go in the biohazard soft. And then other things go in the regular trash. Please pay attention to the cleanup, to the lab cleanup process. Okay, moving on to a new experiment. In this experiment, we are going to learn about how to count the number of RBC. To start with, class, I would tell you what I experienced as a student. When I was a student taking physiology, my instructor told me, okay, you have about 5 million of RBC in 1 cubic millimeter of blood. As a student sitting on the other side, I had no idea what 1 cubic millimeter looks like, trust me. What did I do? I memorized it, got an A in class. Now, as I started working in labs, I realized how small 1 cubic millimeter is. And I would like to share that with you. 1 cubic millimeter is like this much. Do you see the red dot? That's 1 cubic millimeter of blood. And this 1 cubic millimeter holds about 5 million RBCs. So you can imagine how tiny they are. Therefore, what's the point, Rupa? The point is, if you have to count RBCs, obviously you need a microscope, but then you need a tool and you need something that will dilute the RBC cells so they are countable, right? You need to count them, so you need to see them separately. So you need to dilute the sample, the blood sample, and you should be able to count them, right? So that's what we are going to try. We will try to count the number of RBCs using a glass slide, and it's a special slide and we call it hemocytometer. Okay, so what do we do? The first step is obviously you take an alcohol swab and you clean a little portion of your finger. Then you poke yourself with a lance or a lancet and you let the first drop of blood go, and then you you take out a little more blood, and then like we did for the hematocrit, you fill out a capillary here. You fill out a capillary with blood. Now what do? You have to dilute this blood, right? So you take this blood and you put it in this solution. Do you see this solution here? This has a name. Don't worry about it. It's actually called Unipet reservoir. So that is this thing. But for my students, I want you to know the concepts, right? So know that you have to dilute the blood. You have to dilute it so that you can count RBC. That's our goal. So you dilute the blood in this isotonic solution. Why isotonic? Why isotonic? Because you don't want your RBCs to shrink neither you want your RBCs to burst, right? So the cells should be in isotonic solution, so the cell shape remains same. So they are happy. So you dilute the blood in this isotonic solution. And then you take, with this tip, do you see the tip here? You use this tip to put a little bit of blood, maybe a drop or so, into the slide here. Do you see this thing? You can put a little bit here in the V of the slide. Now this is obviously a special chamber that helps us to count red blood cell, and this thing is called a hemocytometer. That's a hemocytometer. So how do you do this? You take the hemocytometer, you clean it with alcohol and lens paper. You also take a cover slip as shown here. You take a cover slip and you also clean it up with alcohol and lens paper. And then you gently put the cover slip on top of the hemocytometer. You are set. Now you take this little thingy, like your isotonic solution where you have the blood dissolved, and you put a small drop here. You can also put a small drop here. It's up to you. So like this. So you are loading the hemocytometer. Do you see how you put the tip on the V and you just put one drop or so? Good. Then you look at it under the microscope. Now I want you to understand one thing, class. We are not staining the blood, right? So don't expect to see a lot of colorful stuff under the microscope. We are not using any stain. We have just diluted our blood and looking under the microscope. So yeah, so don't expect bright red colored red blood cells. No, that's not what we are going to see. We'll just see some pale colored cells floating around. That's it. Okay. So now that the hemocytometer is loaded, we will look under the microscope. Now listen carefully. This YouTube link actually shows you how to load a hemocytometer. Again, a cool video. Hopefully you will learn something out of it. So please do watch when you have time. Okay, now. So you set up the microscope, you pull it out from your little cubbies. Oh my gosh, I miss my lab so much. Okay, so you pull out the little microscopes and you start at scanning power, right? You start at scanning power and look for a cross. That's all you try to find under scanning. Once you find the cross, which is like this, you then go to low power. And this is what you see under low power. You see all these grid lines. Right? Obviously, we are human beings and we cannot count every single tiny square. But we will focus on the 5 smaller squares that are in dark gray. So we will count the cells here, 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 and here. Okay? So this is low power. Obviously, we need to move to high power or 40x in order to see the cells clearly. So this is what we see at 40x. Okay. And at 40x, you should be able to see the 5 dark gray squares. This one, this one, this one, this one, and this one clearly. And we will count. Okay, and you will start counting. So you will start with any one square. And this is how we count. Now, listen very carefully. You might expect one question based on this information, okay? So I will give you a picture with cells like this. You will see the grid lines and the cells. And I will say, how many cells do you see? Count the total number of cells in this picture. So you should know how to count it. Make sense? So we will first look at this side and this side. Okay, we are just looking at these two sides, and please hear me. For these two sides, if a cell is touching the outermost line, that means if a cell is touching this line and this line, we don't count it. Okay, so do you see a cell that's touching the outer line? Yes, we do. So we are not counting this. We are not counting this. Make sense? Now, let's look at, let me see if I can change color. This is so annoying. Let me do it this way. So let's look at the middle line. This one and this one. Do you see any cell that is touching the middle line? Yes, we do, Rupa. This one and this one. So we are counting them in, right? So yes, we are counting the green ones. Okay. Now let's look at some other cell, at the third line here. This line and this line. Do we see cells that are touching the innermost line on the left and on the top? Yes, we do. Do we see cells here? Yes, we do this one and we see this one. Are we counting them in? The answer is yes. Okay? All right. So let's review. When counting, we first start with the right hand side and the top side. Sorry, my bad. We start with the left and top. We see 3 lines on each side, right? If a cell touches the outer line like this, do not count. So cross, cross. If a cell touches the middle line like this and this, count. Yes, yes. If a cell touches the inner line here and here, please do count. Yes and yes. Good? However, things are different for the right hand side and bottom side. So let's look at right and bottom. Things are different here. So let's take a look at the outermost gray line. Okay. Do you see cell touching the outermost line? Yes, this one. Forget it. We are not counting it. Oh, also we are looking at this one, class. Sorry. Okay, are we seeing cells that are touching the outermost line in gray? Yes. We are not counting these. Now let's look at the middle lines. This one and this one. Do we see cells touching the middle line? Yes and yes. Are we counting them? No. Forget. Now let's look at the innermost line. So, oh my god, I cannot draw straight lines on the computer. Let's look here and here. Do you see cells touching the innermost green line on the right and bottom? Yes, you do. You see this one and this one. Are we counting them? Yes and yes. All right. So let's do a quick review, super quick. I have a minute and a half. Oops. Left. So you see 3 lines here and 3 lines here. What's the point? If you see a cell touching the outermost lines, nope, we are not counting these. If you see cells touching the middle line, we are not counting these. If you see cells touching the innermost line, yes, you are counting these. Okay? So practice, and you might get questions on this on your exam.